So today we're going to talk about diseases, and I know in the kind of webinar outline it said pests, including insects, and I have made a last-minute executive committee decision not to talk about insects because the disease talk is about, I don't know, 3 hours. Most of the times I want to give it to Master Gardener volunteers and others, and we just have like 50 minutes. And so I thought I would stick to my strength and just talk about diseases of trees. And so that's what we're going to do today. And hopefully, when you look at trees, you should have a filter for whether or not you think they look good or not. And if you're looking at this tree at the Huntington Library, it's no longer there. They took it down. That should not look good to you. There's clearly something wrong there. And that's what we're going to talk about today. When the trees don't grow right and something is not looking right to you, and particularly whether or not it might be a pathogen related or disease related situation. So in this webinar, we're going to define what diseases are. We're going to give some diagnostic essentials, go over some common disease types in the landscape, the ones that I think most people encounter the most. And we're going to have a few comments about, and they're fairly general comments, how to control diseases, excluding using pesticides. We're not going to talk about that today. But one thing I wanted to make kind of clear is that we can see in this tree growing here in the forest in the Chiricahua Mountains, there's a big canker going from the ground all the way up into the canopy. And this is a madrone tree, Arizona madrone. And, trees get sick on every single part of their being, from the fruit to the flowers, the leaves, the bark, the wood, the roots. Every single part of the organism can get some kind of a disease. And very often they're specific to certain parts of the tree. And so pathogens of trees are common. They're part of the ecosystem, and sometimes they're not. Sometimes they're exotic, which can have really dire consequences for native populations of trees. So, trees can cause loss of function. Diseases can cause loss of function in trees that lead to their failure. And certainly this is a horrendous failure that occurred in Ojai about a decade ago. And these owners were so concerned about this tree, they hired an arborist to get up in there, and you can see he's got this mega extension ladder, and he was inspecting this crown, which is clearly 30 or 40 feet above this building. And they're so worried that a branch would fall, but nobody really inspected the roots, and the failure didn't occur in the branches. It occurred on the whole tree. And so, this is something we have to be aware of. People, I think rightfully so, are afraid of large trees, and particularly when they surround or cover their dwelling, it causes some concern. And so we have to be aware of the kinds of organisms that can lead to these sorts of failures. So disease is an interaction. First of all, it's something that occurs over time. Very often people notice things that have been happening for a long time, and they finally see it for the first time, and they go, wow, look at that. That just happened. But in almost all cases, it didn't just happen. There are latent periods or periods when nothing is happening. There's a time when things first start, and certainly a time when they end, like when the affected parts go through the tree chipper. But diseases can be both biological or biotic, where there's an active pathogen causing the symptoms, or they can be abiotic, where there is no pathogen. And in that case, disease is just an interaction between the environment, the host, and time. But it should be noted that all diseases occur over time, that they don't occur instantaneously, and the time function of a disease might be relatively short, or it might be over decades. There are some of the pathogens we'll talk about today that can be resident in a tree for an awfully long time. So all these factors, especially the main ones, host, environment, and pathogen, interact back and forth with each other. So the environment can be predisposing to the pathogen's development, and it can also be detrimental to the host. On the other hand, the environment might be harmful to the pathogen and helpful to the host. So depending on how these interactions are flowing, this determines whether or not disease is going to occur. And also, this gives us kind of a graphic for understanding how to stop disease. If we can change the environment so that it's less conducive to the pathogen or is more conducive to the host, if we can get resistance in the host to the pathogen so it's less susceptible, if we can eradicate the pathogen or exclude it or keep it away somehow. These are all methods of control. So this is not really any particular data. It's just sort of from my head. And it's kind of diagramming how important different kinds of pathogens might be to shade tree diseases overall. And you see I've got the biggest section of the pie is abiotic, because the non-biological things that happen to trees are so common, especially in the urban environment. And they cause symptoms, they cause abiotic disease. So they're probably the most common thing that we have that causes trees not to perform well. And then fungi would be the next most common. And then that's followed by nematodes, bacteria, viruses, and parasitic seed plants. And these bars are just rough guesses based on my 3 or 4 decades of experience of looking at this stuff. So there's no real data here, but that's just kind of the way I see it in terms of shade tree diseases. So in order to really understand tree diseases, we have to be able to diagnose them. We have to be able to understand what we're seeing. And there are 2 broad categories of this sort of diagnostic essential: understanding what disease does. Disease is a physiological change in the tree that causes the tree to change something so that we can see it, and that's called a symptom. So it's an abnormal change in the physiology and results in some sort of thing. Like in the case of this Raphaelepis, the leaves are turning red and they have brown spots on them. And so those are physiological changes brought on by hormones that are upregulated in the plant that turn the foliage red, and the pathogen killing some of the cells. Now, the other thing that we might see are the actual cause of this set of symptoms, and that would be a sign. So a sign is the actual body of the pathogen, whether it's a fruiting body like a mushroom or spores like on powdery mildew, or hyphae, something like that. And sometimes they're microscopic, like these spores of Entomosporium, shown under the signs column there. You can only see those with a microscope. You're not going to see those with your native eye, your unmicroscopic eye. So a lot of diseases don't have signs that you can see easily. Virtually none of the viral diseases have signs that you can see without extreme magnification. And bacteria are pretty hard to see, so you don't see those. So some of the pathogens have no signs, but most of the fungi do have something that you can see, at least with a hand lens, you can see. I want to talk about some big, broad categories of disease that affect trees all over the country, all over the world. And the first, probably most recognized disease of shade trees is Anthracnose. And you might not call it that based on your own experience. Here we have, 5 different kinds of plants: sycamore, oak, maple, ash, and elm, all showing various symptoms of anthracnose. So what is anthracnose? And it's not anthrax, okay? Don't make that mistake. Anthracnose is a disease of leaves and twigs and small shoots. And you might call it a blight, because that's exactly what a blight is. But in anthracnose diseases, plant pathologists categorize them that way because of the way their spores are formed. So it turns out that anthracnose diseases have all their spores in a kind of structure called an acervulus, which looks like a little volcano erupting out of the epidermis of the stem or the leaf or whatever it forms on, with spores inside of it. And these are splashed around at various times when there's water, and the new infections are timed to the new growth of the host. So this is something we call phenological synchronicity. And the pathogen knows to have the spores ready, formed, ready to release. The acervulus is open right at the time it's going to rain and right at the time the tree happens to be growing. And so anthracnose often shows kind of a bottom-up symptom where the bottom is defoliated and the top is still green. That's very typical. And also the other very typical symptom is for the necrotic leaves to follow the veins, as you see here in oak and also here in sycamore. So different fungi with different names cause these diseases, but the mantra is pretty similar. And these organisms tend to oversummer. In other words, they go dormant. They take a rest, if you will, when the weather gets hot. So very often and typically the anthracnose, unless you're near the coast where it never gets hot, but very typically when warm days and hot summer weather comes, these fungi go into remission. The defoliated tree comes back and it recovers. Most often these organisms are native fungi growing on native trees. And this isn't always the case, because in this one slide, this is Arizona ash growing in California with anthracnose. But the pathogens are common all over the Western United States and in the East for Eastern species. And so the point I'm leading to is that these are adapted pathogens to adapted hosts. They've evolved together basically. And in these evolved relationships between pathogens and hosts, killing your host is not the evolved outcome. So anthracnose diseases typically do not kill their host. They make it look bad. They can cause significant damage, but almost always the host recovers, unless there's some other factor going on. So probably the most common blight disease that people would encounter with trees is fire blight. And this would be in our pears particularly, and in the evergreen or flowering pear the most in the West Coast here. But on apples and on other rose family plants, cotoneaster and such. This disease is so common in California, particularly on Pyrus kawakamiae, which is the quote evergreen pear. And it is something that for many years people have said, you got to cut it out, that we understand, but that you need to also sterilize your pruning equipment. And we had a very famous plant pathologist in California, Dr. Bob Robbie, who taught at UC Berkeley for about 40 years. And every year he would try to inoculate pears with fire blight bacteria with clippers, and never once could he do it. And that's because most of the infections occur in the floral nectaries. Again, there's the phenotypic synchronicity. The bacterial ooze comes out of old tissues in the spring when it's warm and wet. The honeybees pick it up on their legs and they visit the flowers and they drop it on the flower. And the bacteria gets into the nectary and the first infection is in the peduncle. So here you see a bunch of blackened peduncles and moving into the leaf. And it'll move down that shoot and get into larger and larger wood unless you prune it out. So the trick on fire blight is to get rid of it as fast as you can by pruning in the springtime. Another really important category of disease for trees are canker diseases. And cankers are dead spots on a stem. And this can somewhat intergrade with root rot diseases, because root rots, if they grow up off the roots onto the stem, can cause a canker on the lower part of the stem. And so it's a totally different disease category causing this category. So sometimes things are a little bit confusing, but canker diseases simply refer to something girdling a stem, whether it's the main stem or more usually and typically a branch. They lead to a girdle and then eventually a death of everything distal or away from that girdle. And usually we see symptoms early on, such as yellowing or flagging, if you will, in the tree, where branches seem to go off, they turn kind of lose their color, they go dull green, then they go yellow, then they turn brown. And when that's a distinct thing happening at various times in a tree, there's a couple of possibilities. It could be a fungal or a bacterial canker, as we see with Chinese elm canker here, Stegophora, or it could be completely abiotic. In other words, without a pathogen, but with a rodent. Rodents cause an incredible amount of damage. They like some trees more than others, and they'll girdle the branches. And it takes a little while for the xylem to die. And it keeps sending water even though it's been girdled. But eventually, since there's no cambium, it dies. And then you get a very, very similar symptom to a fungal canker. So when we're looking at these kinds of things, when you see initial symptoms of yellowing, maybe necrotic branch, don't just assume it's fungal. Go and look to see if there's chewing on that branch, as that is very, very common. Another thing we get involved with trees, particularly in canker diseases, is looking at the bark and seeing the bark for either being healthy or diseased. And bark has to crack. It has to split apart for the tree to get larger. It can't not split and increase in size. So bark has to split, and it usually breaks or splits vertically, like we see on the left. And that's called a growth crack. And in the very middle of the split is this kind of salmon colored tissue that indicates a healthy growth crack. Now, the other way that a tree could split is from just drying out, where the bark loses moisture because it's dead, and the vascular cambium is no longer feeding a living phloem. And when that kind of drying out occurs, we get horizontal cracks coming off of vertical cracks, as we see on the right. These so-called disease cracks can be indicative of a canker. So a pathogen has killed the cambium under the bark, and both these are coast live oak, so the bark is very thick. But in time, if it's not alive, it will dry out and it will start to crack longitudinally or horizontally. Horizontal cracks on trees are always a harbinger of something not good. Either an insect has been in there and caused damage or cracking, or a pathogen has done the same. The other symptom that we might see with canker diseases, and very typical and common, is bleeding. So trees will ooze sap when they're being attacked, and this is a defense response. It's a typical defense response to insect injury, but most people may not realize it's also a defense response to fungal or oomycete, that's water mold attack. And that's what we have on the two end images, is Phytophthora and bleeding associated with those, and a particular canker here on the silver maple. And in the middle slot, it's cypress canker and bleeding affiliated with that. If we look to the inside of something we don't think is healthy, and we find necrotic tissue, or oxidized tissue, this is always consistent with our oomycete pathogens, the Phytophthora, and they're the so-called water molds. And when the cankers are low to the ground and apparently coming up off the roots, this is very likely the cause of the disease. There's other diagnostic or confirming things we can do. There are test kits that can test a little bit of the tissue, much like a pregnancy test kit, dipstick kind of thing, and can confirm Phytophthora, or tissue can be sent to a laboratory for isolation and identification of the pathogen. In the case of these pathogens, which I'll tell you a little bit more about in a few minutes, there are no signs. There's nothing you can see here except a different fungus on the palm here, pink rot growing on the outside. But the Phytophthora can't be seen because it's intercellular. So as cankers go along, they keep killing larger and larger pieces of wood. And when left too long, they'll kill major portions of a tree, as we see here in this ficus tree in Beverly Hills. It's lost a lot of 2/3 of the canopy to this pathogen. So cankers are progressive and they work backwards. If they start on the edge, they'll work back down into the canopy of the tree, killing larger and larger stems as they go. The other thing I want to say is it's not always obvious that something is diseased. Like in the upper photo here, it's obvious to my eyes because I see the horizontal cracks. Okay, you can see them right there and here. You know something is wrong here. But then when you wash the dirt off this ficus, you get a very much clearer view here. In the lower picture, this is brown and this is green still. So there's something wrong here. And you go in a little bit closer and you see, okay, yeah, we have the horizontal cracks showing that this is not healthy. And also there's this black surface, and looking even more closely, it's covered in little black dots. And those are the fruiting bodies of the canker fungus. So we think of fruiting bodies as a mushroom or something, but this is just a little receptacle that has spores inside of it. But microscopically speaking, there are probably 1 million spores here in this image. So each little receptacle has many, many, many spores in it. And when the time is right, when there's water and these spores are ready to go, they'll release into the environment. And so this is the disease caused by Botryopsera, and it's very common throughout the world. It's not just a California thing, it's an everywhere thing. And like a lot of fungi, this pathogen makes 2 different kinds of spores. It makes a kind of spore that comes from mitosis, or just cellular division, on the left, called conidia. And then it makes another kind of spore called an ascospore, which is a result of sexual recombination of different strains of Botryopsera. So the ascospore is very resistant. It usually is the first infection, and the conidia are formed in other fruiting bodies throughout the growing season and continue to infect plants. So this is the same mantra that many different kinds of fungi use. They have an asexual spore for rapid spread and a sexual spore that's often the primary or the first inoculum. So with canker diseases, much like with fire blight, the first thing to do is informed pruning. And by informed, I mean knowing how to prune, which we covered in the last or webinar before last. And the main thing is to realize that the collar, or the shoulder of the stem here, where the branch comes into the main stem, is a defensive zone. And you can see where the arrow is pointing. The discolored wood is trying to advance into this area, and the tree is essentially keeping it from progressing further for a time. And this is, like the progression of a huge army. I mean, in time, if there's enough energy in this dead branch, there's enough fungal biomass, it will penetrate this reaction zone and it'll get through to the tree. But what we don't want to do is to cut behind this collar. If we made our cut back here, we would take away the plant's defenses to prevent these organisms from getting in, and then it would just get worse. So when we already have disease in a tree, such as here, where there's a cankered branch dying, you definitely want to just cut outside the protection zone. Even though you may not get it all, that's okay. You leave the plant to deal with it, and you don't breach this defensive area that trees have. So powdery mildews is another big category. There's probably a powdery mildew for every plant on earth. We just don't know where it is. It's probably somewhere. and they get on a lot of plants. They cause damage on a lot of plants. And the damage of powdery mildew, at first it may just seem like, oh, it just covers the leaf. It looks superficial, but it's actually very distorting. And I show some extreme examples here. The brooming here in coast live oak, causing hypertrophy and hyperplasia, overproduction of shoots, and stunting of the leaves, and it's completely covered in spores. The distortion on sycamore here, twisting and folding this leaf, it can't develop. Euonymus is probably pretty indelible, but this infection likely happened on a leaf that was already formed, so it's not distorting it too much. But on rose, the leaves are distorted and the flowers will not form and be distorted as well. So the big problem with powdery mildew is distortion. And powdery mildew is common on some things more than others, like sycamores and crepe myrtles, very, very common. And there is resistance. The varieties of crepe myrtle with a Native American name on them usually are resistant to powdery mildew. And there are just other trees we can plant that don't get powdery mildew as much. Powdery mildew also is an unusual kind of pathogen called an obligate biotroph. Obligate biotrophs are fungi that have to be on a living plant. They cannot complete their life history on dead material. They don't survive in mulch for very long, although their sexual stage does occur on fallen leaves once the conidial stage, which is the mildew stage, is finished. So everything you see on the screen here is conidia or asexual spores. And this is why powdery mildew is so successful. It is able to produce a lot of spores and infect new growth all season long. And when the leaves get burned out and used up, the whole thing falls to the ground and it converts to a sexual stage. And those sexual spores will sit there and wait for rainfall and get splashed up onto new growth again in the new year. So powdery mildews are best tackled through resistance. And if you're going to use a pesticidal approach, doing prophylaxis or a covering of fungicide early before the infection occurs, but mostly just getting a resistant variety is the best thing to do. Now, root rots are a big category. They're the thing I tend to see the most killing trees. And we have to be cognizant, as I said, that disease can occur on any part of a tree, from the flowers and fruit to the root tips. And when disease occurs on roots, if it's particularly virulent or conditions are really favoring the progress of the disease, then the host, the tree, can really suffer. And the two big pathogens that we have, at least here on the West Coast, are Malaria, which is the oak root fungus, and Phytophthora species. There's several, both causing root rot of many, many hundreds or even thousands of different landscape plants, including trees. So the other way that we could have root rot is through abiotic means, by flooding, natural gas leaks in the soil that push all the oxygen out. And I've put fabric sheet mulching. My colleague Linda Chalker-Scott is on a tear trying to renounce sheet mulching for having no merit scientifically and actually being harmful because it impedes gas flow to roots under the sheet mulch. So I don't think we have a lot of evidence showing that weed fabrics and sheet mulching kill roots. I don't think that's the case, but they may be contributory to abiotic conditions, especially when flooding is present. So something we need to know about root systems is that they naturally harm themselves. Roots are not formed like shoots. Yes, Tracy.
You might also add that sheet mulching can interfere with water infiltration.
Exactly. And great additional point. Especially on new or young plantings, you risk basically watching your plants dry out and die if they've been sheet mulched. So back to how roots form, shoots are very pre-programmed. They have buds, they grow sort of naturally and in preordained ways by the genetics of the tree. There's a phyllotaxy or an arrangement that's associated with every taxon. Your hand is still up, Tracy. Does that mean you have another question?
No.
Thank you. Okay. And roots are not that way. Roots are not formed so much as birthed. They literally erupt through the cortex of the young root. And here we see the sequence of the young pericycle, and there's this little red zone. It's building up to make a branch root, and it's getting more intense, and it eventually forms inside of its own cortex. It's literally giving birth to its branch root. And when the branch root pushes out, it breaks the epidermis open. There's loose cells, there's cell solutes, there's all kinds of things happening here. And this is a very beautiful opening that a plant pathogen could enter. It's the perfect way to get inside the root. The plant has created its own injury for a pathogen to get into. And so just by the nature of the way roots form, they're self-injuring, and there's blood in the water, so to speak. More like, I guess we could say there's exudate in the soil moisture. And that exudate is going to be stimulatory and attractive to some pathogens. So let's talk about root rots caused by water molds briefly. Water molds are not fungi. That's the first thing you should be aware. They're like highly evolved algae that don't have chlorophyll anymore. And they are very common. Phytophthora is worldwide. We think about 500 species, and I'm not sure how many have been found yet, maybe around 200. They're highly evolved. They have no saprophytic ability. They're strictly parasites. And when they're not on or in a host, they reside as spores in soil. They're common in horticultural industries, especially nurseries and landscapes. They're microscopic. There are no signs and only symptoms. And so this is a eucalyptus tree growing in Ventura, California, and it's not a kind of tree that gets brown leaves. So you can see the whole tree has died. And this is an important symptom for understanding root rot. Very often, when you see a root rot affected tree, the entire thing has been affected, not a branch here or there. The entire thing goes off. It turns color, it changes, or it outright dies suddenly. And that is just a harbinger that you have root rot, and particularly caused by oomycetes, although also by oak root fungus. So here we see some of those color changes. Bradford pear on the left turning red, and a healthy one in the same image. So it's not a fall kind of thing. It's not fall color. This is turning red in the spring. It shouldn't be doing that. And in the picture on the right in Kiev, Ukraine, boxwood shrubs that have gone off and turned straw colored or brown. So both of these cases are Phytophthora, where the root systems of the individual plants are being completely destroyed. And like the other symptom slide I showed you earlier, if we get a pocket knife out and look beneath the stem of these kinds of plants, you will often find the necrotic tissue typical of Phytophthora. So in diagnosing these root rot diseases, really a pocket knife is very, very handy, or a sharp dagger or what have you. I wouldn't get out your best kitchen knife, but usually a very stout pocket knife is helpful. And you might go, well, what if I'm digging around in a healthy tree? Well, that's not good. So you always look for outward symptoms such as bleeding or cracking. And if you see that, then you can go start to dig around a little bit with your knife. And if it's brown under there, you can keep digging. You're not hurting a plant that's already basically dead, as all of these are. Even though they still have healthy tissue above, if the base has girdled or is significantly been attacked by the pathogen. Usually they can't be saved. So Phytophthora, as I said, is a water mold and it has a fairly complicated life history. It survives in soil as chlamydospores, which are these dark things here. And it waits for a host and it rapidly forms something called a zoosporangium here. And from that zoosporangium, swimming spores are initiated that move through the water, soil water, and they're chemically attracted to the leaking root. Now here we see a root with root hairs. Most trees don't have root hairs because they've been replaced by mycorrhizae, but they will have branch roots. And where the branch root is popping out, that's where you'll have the zoospores try to attack. The other place they attack is right near the root tip, near what botanists call the zone of elongation. So once they get to the root, they pull in their whiplash and tinsel flagellae, and they attach themselves as another kind of spore called a cyst. And that spore germinates and grows on the surface, and it forms a holdfast structure that allows it to penetrate, and then it dissolves the cell walls and goes inside and starts to cause the rot of the roots. And so these pathogens typically mass attack. So here you see a root tip of avocado with several hundred zoospore cysts on it. And when there's a lot of Phytophthora in the soil, a susceptible root usually cannot survive that onslaught and it will be killed. So the other big organism we have in California and throughout the United States is oak root fungus or Armillaria. Most of the time it's Armillaria mellia, but in other higher elevations, it could be Armillaria ostoyi or various other species. There are different species and what mycologists call biological species in the Armillaria genome. And it's actually under a lot of changes and names are changing. So right now, Armillaria mellia is a good name that describes a lot of the oak root fungus we see in landscapes around the Western United States and probably other places too. These are worldwide pathogens. There are armillarias in the tropics, in the East Coast, North, South, both hemispheres. And the pathogen is a mushroom forming organism. And it survives by pathogenically attacking trees. And here we see it taking out its preferred host in California, which is the Peruvian pepper, Shinus mole. If you see a dead Shinus mole, it's almost certain that Armillaria killed it. And this is what's called the Mycelial Fan, or the mycelium of Armillaria growing under the bark of a Shinus mole tree. So I've taken my knife, I've removed the outer bark, and it reveals the mycelial fan. You see something like that growing where the cambium of a tree should be. It's almost completely certain that it's Armillaria, although there are a few other things, jack-o'-lantern fungus and a few others that could cause similar fans of mycelium. And here are a couple of my colleagues, Farm Advisor Emeritus Don Hodel on the right, and City Arborist here, Nathan on the left, and this is in Ventura. Nathan is in a different city now. I think he's in Santa Barbara, Nathan Slack. And the tree is Ficus Microcarpa, and the mycelial fans have gone above head height on this large specimen. This is the tree overall. And you can see, despite the extensive infection, it still has leaves, probably because the oak root fungus hasn't girdled the whole thing yet, but that it's off. This part of the canopy is not right. It's not as green. It's turning brown. And the entire tree is, I would say, not as vibrant as a Ficus microcarpa could be. And so the symptoms can be rather subtle in the canopy, while at the same time, the signs on the main trunk can be quite extensive. So the pathogen has to girdle the entire stem before the tree will die. And even then, it will take some time for the xylem underneath the fungus to die. So water can keep going up that tree for some time. And then eventually, if it is girdled on the main stem, it has to die. It can't come back if there's no vascular cambium. And so we know from research I did over the years in Ventura County on survival of pathogens in yard waste mulches that Armillaria mellia is absolutely annihilated by trichoderma. Trichoderma is this green thing in the Petri plate. This is a Petri plate where I was trying to get pieces of Armillaria that we had buried in mulch piles to grow and see if they were still alive after a certain time. And we were able to show in that research, we repeated several times, that just within a few days, almost all of the mycelium of Armillaria becomes colonnaded with trichoderma. So trichoderma is ubiquitous. It's not something you have to buy. It grows on wood. It's a saprophyte, but it's also a hemiparasite. It likes to parasitize other fungi. And there's two things that make this biological control work. One is taking the inoculum and chopping it up. In other words, running it through a chipper. That's the first part. You have to break it out of its contained environment. And what Armillaria tends to do is it puts a pseudosclerotial plate barrier over itself in dying roots in the soil, so that it's sitting there impervious to everything, even trichoderma in the soil. But then when you break that open, it allows these fungi to get in there and start to eat the Armillaria. And so disturbing the soil, it turns out, is a critical way to be more successful in replanting trees in Armillaria infested ground. And the other way that trees die of root rot is from anoxia or hypoxia. And this is usually due to suffocation. Well, how does that happen? The most common way I've seen is natural gas leaks. If there's a natural gas leak in a soil, it pushes out all the oxygen, and the roots that are exposed in that environment can't respire and they die. It also does another thing to soil, which is really interesting. Because there's no oxygen, the oxidation reduction state of the metal elements in soil all change. So copper, manganese, magnesium, sulfur, which is not a metal, but it does this, and nitrogen, they all are reduced. So they change their oxidation state and they change their color. So sulfur goes from something like sulfate to sulfur oxide and sulfur dioxide that smells like rotten eggs. And generally these anoxic soils smell badly. And if they do smell badly and they are colorful, that is, they have these dark regions where iron has changed its oxidation, and manganese and magnesium, they've all changed. They create compounds that are toxic to roots as well as the gases being toxic to roots, as well as the lack of oxygen being toxic to roots. So all the way around, this is a toxic situation. And I don't think we approach that to any great degree in compacted soils and even in flooded soils, because they have to be completely covered in water for a long time, a matter of weeks before this happens. Now, we have seen this in agriculture in Ventura County in years when it actually rained. What's that? 2005, I think, was a good one, and maybe 2015. But I seem to recall in 2005, the lemon orchards being underwater for a number of weeks and trees just dying from suffocation. Okay, vascular wilt diseases are another big category that particularly are common in the east coast of the United States with verticillium. Verticillium wilt is a big problem there. We have verticillium wilt in the west as well, but not as much as they do in the east. The symptoms will be like canker diseases where branches die, but there's no girdling of the branch. The girdling is happening internally in the xylem that's being plugged up with the fungal organism. So the symptoms may or may not be obvious, and some plants can live with a low-grade infection of wilt disease and just not grow. They just sit there. Others get severe disease and start losing branches, or the entire plant dies. Curiously, in our study of climate-ready trees in California, we're seeing one of the trees that's done very well in Arizona, which is the rosewood, getting wilt disease in California. So I want to talk a little bit now about disease control approaches. In other words, okay, I've identified a disease, I've looked at the symptoms and the signs, I've done my research, I've figured out maybe what it is, maybe I talked to my extension agent, and I kind of have a clue, but now what do? I want to get rid of the disease. You know, it's a great world if we could just do something and eradicate it, like in medicine. You know, you take a pill, it goes away, right? Unfortunately, that's rarely the case in medicine, and in landscape diseases, also rarely the case. A lot of times it means getting rid of the diseased plant, which is exclusion. Taking that plant, or roguing, eradication, getting that plant out and removing it from the environment so other plants don't catch whatever it has. Exclusion really refers to not ever getting it in the first place. So we know that certain diseases are coming toward us, like laurel wilt coming across the southern United States toward California. And we're trying to keep it out of California by not having people bring firewood into California or move the insect pest, which distributes that pathogen. So exclusion is a big one. Quarantine helps exclusion. In other words, having a quarantine on an area and not allowing plants to move in or out is an attempt to exclude pathogens. Of course, chemical control is the one most people would probably think, you know, where can I get a fungicide to control that disease? But chemical control is often not eradicative. It's often preventative, or it prevents spore formation, or it prevents penetration into the plant. It prevents the pathogen from further developing in the plant if it's systemic, but it often is not eradicative, i.e. killing the pathogen in or on the plant. We do have some eradicative fungicides, but be careful with those, because very often things that eradicate the pathogen also kill the host cells. So a consequence of eradicative fungicides is phytotoxicity. And the plant dying where you sprayed the pesticide indicates that you've killed the pathogen there. And this is especially the case with biotrophic pathogens like powdery mildew. If you use an eradicative powdery mildew fungicide, you're going to have phytotoxicity, because it literally kills all the cells that the powdery mildew is involved with, which can be quite extensive. So this is why chemical controls, whether they're systemic, prophylactic, or eradicative, need to be put on early to limit damage to the plant and try and stop the progression of the pathogen early. And that just requires a lot of knowledge. It requires you to know what your pathogen is, what it's susceptible to, fungicide-wise, and when it infects, so that you know when to get in there ahead of it. Biological control is using living agents to limit, interfere, or interrupt the progress of a disease. And I'll talk a little bit more about that in the next slide or two. Resistance, of course, is getting the host to resist the pathogen, either all or none or some, and allowing you to either grow a crop or produce what you need to do and survive the infection. Yes, Tracy.
While you're speaking on resistance, can you also speak on Armillaria cultivar resistance in plants? Is there such a thing in your opinion?
I have not seen anything like that. I think we see species differences. Like, for instance, a species of oak might be more or less resistant to Armillaria, but I don't know anything about cultivar resistance. In agriculture,
That may have been a bad choice of words on my part, but just in general, is there resistance among any trees that you're aware of to specifically Armillaria was the question posed.
yeah, that's hard to get at because resistance is, it's a very genetic discussion. So what you're really asking is, um, is there a genetic response in the host to the presence of the pathogen? And Armillaria seems to be so broad in its attack. It it's not like there's a way for a host to prevent it from happening. So I don't think there's a lot of resistance. But if we were to get Dr. Baumgartner on the line, she might tell us that some varieties of grapes might be more resistant than others, because that's what she's working on is grapes. And it wouldn't surprise me that perhaps some vines, some cultivars of grape have more phytochemicals or are less attractive in some way that Armillaria doesn't occur. So specific resistance like we have in snapdragon rust, where you have all or no resistance to it in different cultivars of snapdragon, that's conferred usually by one gene. And I don't know anything like that that's going on with Armillaria. I think most of the resistance, if it's there, is going to be multigenic, and it's going to be across many different pathways. Maxwell, I did see your hand go up, but we'll get to you later when I finish up. Okay, so we have cultural control, which is a good example here from Dr. Baumgartner. She's excavated the root junction because all these grapes were planted about 12 inches too deep. And that ended up slowing the progress of Armillaria in this case. And finally, integrated control is using everything you've got to try and stop a disease. I want to say something on sterilizing tools because it keeps coming up all the time. Should we use Lysol? Should we do alcohol? Should we use household bleach? There's several pitfalls here. First of all, those chemicals that I mentioned are not really registered pesticides. And lawfully, you're not supposed to use them that way. Although people do, people are always on about sanitizing their pruning equipment. And I tend not to sanitize very much. If it was a wilt disease like verticillium wilt, like we're seeing in our rosewood trees in California, and I was pruning from tree to tree and I was going from a diseased tree to a healthy tree, then this might come to my mind because I know that some of these wilt diseases and Botrysphaeria particularly can be moved on pruning equipment. And I did some research a few years back on Fusarium wilt of palm and was able to show that 15 to 20 seconds with a torch on your saw blade will take care of 99% of everything and like 100% of Fusarium. So using a flame is a very effective way to kill things that stick to your saw blade. It's just that most of the time you probably don't need to do this. Some of the time when you're working with pathogens or you're working with diseased trees and moving to healthy trees, then this is a time when you want to think about that. I'm not going to go long on this because it's its own whole 3-hour thing, but fresh tree trimmings mulch, also known as green waste, can help control shade tree diseases. And we know that it helps with root rots because that's what I worked on my PhD on. It's very destructive to the oomycetes to have fresh wood chips on the ground. But I think that it can have help in other ways too. When you mulch trees, you protect the soil surface and you cut down on salinity buildup, and trees stay more evenly moist. There's less swings in the irrigation system, the drying and wetting cycles. And so basically mulches cut stress. And then the other thing they do is they increase roots. Mulching always leads to more roots. And the more roots a tree has, the more of a buffer it has from stress. And this translates even into our canker fungi. So you could make the argument that mulching with fresh tree chips is going to help with canker diseases. Not that it's going to directly impact the canker fungus that's up on a branch, but it's going to improve the resistance of the tree overall, decrease the stress of the tree, and canker fungi are so stress related. They tend to attack trees that are under stress. Finally, I want to emphasize that when you do mulch trees, to use fresh material, as we talked about in the last webinar, because this has the carbon that's available for microbial work, and that substrate is going to encourage a lot of beneficial organisms. And so what does that mean, actually? There are these various methods in which a substrate like mulch can support biological control in the soil. The organic material of mulch is going to let organisms grow that are saprophytes, but sometimes they're hemiparasites of fungi. And so we can get this hyperparasitism where they directly attack the pathogen. And they wouldn't be there if they didn't have the carbon source of mulch to build up on. This would be like trichoderma. And then a lot of these organisms create antibiotics. And so you have the fungi growing in the mulch, but they're also creating antibiotics that are regulatory or harmful to Phytophthora. And some of the microbes are just breaking down the wood, and they're going to produce enzymes that dissolve the pathogens in soil. Finally, the mycorrhizae, particularly the ectomycorrhizae that create a barrier on roots to pathogens, are also reliant on the organic layers in soil that are up on the surface. The hyphae are growing in the organic matter and on the tree roots. And so there's a connection, a direct connection between the organic matter and the tree that is promoting its health. And so in that way, again, the whole concept of litter fall, mulching, fresh organic matter on the ground is promoting the health in all these mechanisms. So finally, I would say that we need to be really aware of predisposing issues. A lot of times, a particular pathogen might be resident, a particular host might be there with it, but the pathogen is not causing disease. And then something changes. We do something different. We add soil or put in volcano mulching, or we take away turf or add turf. We change the environment somehow, and it becomes stressful to the tree and advantageous to the pathogen, and then a disease starts to happen. And so we have to be aware of what those predisposing factors are. A lot of times it's over pruning. Over pruning is debilitating to trees, and it puts them off their energy. They don't have enough energy to make defense compounds, and they become susceptible to pathogens. A lot of times it can be other things, the other plants we've planted, or the combinations, or the irrigation regime, or whatnot. Sometimes it's just the selection or adaptation of what we've chosen to grow in an environment. And so that's shown here. And my last slide, this is a slide from the climate ready tree study in Riverside of two different kinds of oaks. One is the island oak, Corkus tomentella, and the other is the escarpment oak, Corkus fusiformis. The island oak grows in the Channel Islands, Catalina Island, where it never gets particularly hot. Last year, we endured in Riverside 120 plus degrees, and all the island oaks were fried on the south side where the sun was exposed to them. In the same test plot, virtually right next to this tree, the escarpment oaks had no damage whatsoever. And these are all just on their own irrigation now. There's no irrigation even being applied. So they're drought tolerant, but they're not necessarily heat tolerant. And I think this is a really good example. The escarpment oak from Texas is just a lot tougher in Southern California, particularly Riverside, than our native island oak from Catalina Island. And just because a plant is a native plant, if you put it in a non-native setting, don't expect it to endure climate change necessarily. So the kinds of changes we're seeing, particularly in temperatures, drought, and rainfall amounts, is going to drive selection processes. And it isn't always going to be the native plant that we choose. It might be a native plant of Texas or a native plant of Asia, or somewhere where it's more like the climate that we're developing here. So in conclusion then, do your diagnoses, do your research, monitor the symptoms over time to see how the disease is progressing. Devise a control strategy based on all of your research, and avoid poorly adapted plants. This is really the key thing. And unfortunately, this was an easy thing to say in the past, and now it's not so easy because the environment is changing in ways that we don't quite understand. And the adaptations of things that are even native in our own area may be being pushed out. I'm seeing this with coast live oak. Coast live oak burned up like this too in that 120-degree temperature, and it was growing in its native habitat. And so what we're seeing then is just scenarios where plants can't endure some of the changes. And so what is an adapted plant? Well, that seems to be a changing thing. And it's going to be important to choose plants, and particularly trees, that can endure the kinds of changes that are coming at us. Now with that, I'm going to stop sharing. And I see we still have 182 participants. Thank you, everyone. Tracy, why don't you fire away with the questions? Maxwell Norton actually had his hand up, so.
Maxwell included a link that I'm investigating regarding potential Pacific Northwest or malaria tolerant plant species.
Ah.
And the list includes resistant incense cedar, larch, Scots pine, smoke tree, ginkgo, big leaf maple, catalpa, Japanese maple, Japanese pagoda, locust, haha, madrone, mulberry, haha, tulip, shade master, locust, sweet gum, haha, sycamore, haha.
Okay, so what you've just elucidated there is a list where we have
pines,
seen several of
arborvitae,
those
cedar.
plants have armillaria.
Right,
Right?
I'm laughing, yeah, because I have several of those on my own property, and my property struggles with armillaria.
Yeah, the problem with the armillaria resistance lists is that there's always like, I saw that tree with that pathogen. So they're not very good, and they're certainly not. It's like if you see a list of resistant plants, you want them to be resistant, damn it, not exceptional.
Yeah.
And that's not the case with those lists. They're almost always not good.
So, maybe investigate further, use with caution, but realize that the science was not necessarily available when some of these reports were published. And also what we have found through investigation with Dr. Linda Chalker-Scott of the Garden Professors blog and Dr. Downer's association with that blog as well, that much of what has been published, even among cooperative extension agencies, is not peer-reviewed and thoroughly researched information. Much of it still remains anecdotal.
Well said.
So, questions. I'm going to go to this one because I thought it was really interesting and it relates very much to what you just said. So you mentioned how harmful overpruning can be. Yes. Is there anything that you can do when the overpruning is due to a severe storm damage? We had a major ice storm last October in Oklahoma. Trees were leafed out already, and many of them lost 90% of their canopies. We had a similar thing occur in Pasadena a few years back.
Only it was wind.
It was wind.
But no, I feel for you on the ice damage. You just do the best you can. And I guess my guidance would be, you have an 8-inch branch, it's broken, it's shattered. I guess don't cut it off at the attachment. Leave the stub. Go ahead and leave the stub, let it branch, let it shoot out with epicormix, and then it's going to be a slow time with frequent prunings in subsequent years to get it back. But that's all you can do is repair damage, and don't necessarily cut all the arms off at the main stem, because then you're just introducing a lot of decay into the main stem. That's not a good thing. Okay.
Okay, sorry, let's see. So I got this a couple of times and I answered it once. So let you answer this more scientifically, perhaps. Can you replant a tree in the same spot where the previous tree died from a root rot caused by a pathogen? Of course, that depends on the pathogen. Your turn.
Can you say that again?
When can you replant
Oh,
into the planting space where a specimen died from a root
yeah.
rot?
Well, I guess it depends on the root rot, right? Depends on what the pathogen was.
Can we
And
assume
then,
Phytophthora first?
yeah, okay,
That
if it
was an
was
earlier question.
if it was Phytophthora, we do have trees that are less likely to get it and more likely to get it. So maybe not go back in with the same thing that died there. And then think about what the conditions in that site were there that led to the Phytophthora. In other words, how good is your drainage? How much, what's your soil type if it's texturally very fine? And there's fungicides for Phytophthora. We can go in with prophylaxis to prevent infection, and you can up the calcium in the soil to help prevent infection. So there are things that you can do. Now, if it's armillaria is surviving by root chunks with armillaria in it. And so getting out the old root system of the inoculum that caused it in the first place is the key thing. And that just means massive digging and soil disturbance. And so those are the two approaches. Yep.
But that buys time.
Buys time because ultimately the root could explore and find something that you didn't disturb, and it can be infective.
Well, and there are residential properties that are linked, and your tree's roots don't know necessarily property line boundaries.
No.
So what you do on your property, you know, is great, but what is occurring on your neighbor's properties may also affect your property. So also you might mention that the calcium type may be important.
Okay, so when I say calcium, it's the calcium ion, Ca++, but that can come attached to something that causes an alkaline reaction in soil, or something that causes a neutral or no reaction. So if we apply limestone, you're going to make the soil more alkaline. If you apply gypsum, it's pH neutral. So in the east where it's acid soils, go ahead and apply lime. It can't hurt, right? You're just going to move the pH closer to neutral. In western soils, we want to use gypsum for root rot control because it's not going to make our soil more alkaline.
Somebody would like to know what the tree at the Huntington passed away from. Poor thing. Aw.
Oh, you would like to know that, would you? Well, I think, and it was a little ahead of its time, it was what we called it in the day, Polyphagous shothole borer, which we now call invasive shothole borer. And the concomitant fusarium infection. And that was an English oak, by the way, which it turns out to be very susceptible to that pathogen. Our native oaks seem to be less susceptible to it, but that's what I think it was. It also had armillaria.
So it had a multifactorial issue?
It was a disease complex.
A disease complex.
That's when we have more
that.
than one pathogen going at it at the same time.
So this is great because this is the next question. Do fungal infections in trees tend to represent multiple generations and or multiple infections of fungus or one persistent fungus individual within a tree? Thank you for that interesting question, Michael Cumberland. Oh, I'm sorry, Chamberlain.
Yeah, Michael Chamberlain. Yeah, good insight. Yeah, you know, sometimes it's overwhelmingly one thing, like with, you know, Peruvian pepper and Armillaria amelia. You see a dead pepper, you see it full of fungus, the same fungus. It's pretty much that's it. That killed it. End of story. But that doesn't mean to say that some other trees might not suffer all kinds of pathogens at the same time. One of the classic ones that we don't know a lot about is the interaction of nematodes with root soil fungi. And so nematodes will wound roots and they'll cause things like fusarium to get in there. And fusarium is not a very common tree pathogen, but we don't know, for instance, the interaction between verticillium and root knot nematode. That would be a fascinating PhD study for somebody. Please sign up today. Let me know what you find out. But sure, we have all these different kinds of organisms, they interact, and the tree only has energy for so many phytochemicals to make, and it's eventually going to run out of energy. And which one took it down? Probably all of them. And the one that you noticed is going to get the credit for killing the tree.
So Michael Chamberlain also asked earlier, and I responded to, but you might have other thoughts on giving your response about the flame sterilization of cutting tools. He's asking about portable UV wands that have been popularized for sterilizing surfaces based on COVID. Is that something that we could use for pruning tools? I said, I don't think there's research on that, but that's an interesting idea.
Yeah, very interesting idea. And it's, a plumber's torch heats up metal to lots of hundreds of degrees is real fast. But for ionizing radiation and UV light to kill pathogens, it's going to have to penetrate through, debris. And I just think it's going to take a long time. When it's hitting a bacterium on a surface, I mean, it's like the light is hitting that organism straight on. That's relatively quick. I can see that in labs or food preparation or the outside of food or something like that. But not so much when you're trying to kill fungus inside of the debris inside of your saw.
And you're
I think it's going to be less effective, but Tracy's right. I don't have research to cite on that, but it would sure be fun to do and easy to do. I mean, we get some pathogen, we cut it with the saw, we expose it to the light, we plate it out, bam.
I've been hearing information anecdotally from qualified arborists that they are using some of these heat and infrared treatments on living or active infections such as our malaria. So that research could pose to be very interesting in the future. Let's see, the next, something I wanted you to clarify because I'm not sure you did. You were speaking about the flame sterilization. Did you make the point for our lay audience that that is on hand tools and not on chainsaws necessarily?
Yeah, we don't want to combust your chainsaw. So please don't set the oil on fire on your chain and maybe set the gas in the chainsaw on fire. That would be a bad thing. So yeah, hand tools.
And the chain's running through the chain oil and there's limited research that, that can be affected, correct?
Yeah, the initial work was by Howard Orr in the 1970s, and he plated out spores from chainsaw oil and found that it could get in there, and that's where that whole came from. But I don't know of anybody else who repeated that or did it since. So I guess anecdotally, we can go down the anecdotal path, Tracy, and say that we've seen lots of cases where Joe Blow tree schmuck service was pruning, infected palms and then went somewhere where they weren't and suddenly they were with their chainsaws that were dirty. And so anecdotally, we see a lot of that, but nobody's done the science on chainsaw spread that I know of.
Another great master's or PhD opportunity.
Another great study, yes.
So this
come to Cal Poly. We'll get you signed up on a research project.
Yeah, and he will give you like post-traumatic stress. I can just guarantee it. Fresh versus non-fresh wood chips. How old are chips before they are no longer fresh? And non-fresh defined as compost. Are non-fresh defined as composted chips?
That's the way I like to look at it. Yeah, fresh is, it hasn't gone through a biological process like composting. So if it still looks like wood, if you have a handful of mulch and you see wood in the mulch, then it's considered fresh. I did do some research on fresh versus sort of not fresh yard waste mulch. In other words, yard waste that have been sitting around for a while. And it has less biological activity. There's less actual usable carbon in it. And so it's doesn't get as hot when you repile it, and it doesn't control things as well in
But
terms of controlling pathogens.
you don't have like a, 12 days under 180
No,
degrees and that's it.
no, I mean, 3 days after it's been in a guy's chipper box, it's not fresh.
No,
And it's
but
going to be
where is
full of
the biological,
aspergillus and other stuff, but
where's the biological reduction in benefit? Where's that line? Do we know
I
yet?
don't know.
And that's going to depend on feedstock likely, right?
Right. And handling and time, and just keeping freshly chipped trees in a box for a week is a significant degradation. I don't know how significant, but there's less carbon left in that tree box after a week than when it first went in there. The best is to get that stuff out of the chipper on your landscape the same day. That's the best of all worlds right there.
That's where lots of young bodies and students and grandchildren come in. Yeah, because that's the easiest way. In planning for the future warmer environment, do you choose a tree for your own? How do you choose a tree for your own area? One that might last 100 years. Ooh, good luck on that. We don't know.
Yeah, I think you look for the tough trees. I think you look for things that are just bomb proof.
That have
And
ranges,
yeah, ranges of bomb proof. And unfortunately, things that we think are bomb proof today turn out to get something tomorrow that we didn't know about. So there's no guarantee going into the future, but generally, if we look at climates that are 5 to 10 degrees hotter than where we are, then you're going to be planting something. And as long as it can endure your cold temperatures, some trees that are adapted to heat aren't necessarily adapted to surviving really cold climates. So you have to work within the range, both positive and negative, to get a tree that will be adapted. So yeah, that's a tough call. I mean, I don't have a real good answer for you. And also I don't know where you are. The geography changes the decision. So what will grow in Colorado and Denver isn't going to necessarily grow well in Las Vegas, even though the extremes can be similar in some ways.
Or Imperial County in California. That seems
Correct.
to be the edge of some really dramatic changes. This is the last open question. I have a silver maple in my yard that has leaf scorch, not stating whether that's Xylella. Will
Let's
antibiotic
assume it
treatment
is.
control the disease? Yes.
So they would if they could get to it. And the problem with all wilt diseases and scorch diseases, and let me briefly catch everybody up, leaf scorch is caused by a bacterium, and it's vectored by an insect, a leafhopper usually. And leaf scorch is hard to control because the bacterium propagates inside the xylem tubes and plugs them up, so water doesn't flow very good there. So it's hard to get antibiotic there. If you could get it into the system before the system is plugged up, then you would have better luck at controlling the disease. And so it's tough. Once you have symptoms, you're already too late.
So are you advocating for a prophylactic off-label application of antibiotics to trees to prevent a future infection?
Never. That would be against somebody's pesticide laws somewhere.
So you would have to really investigate the pesticide laws for your area to determine if a prophylactic treatment can be applied legally to your trees, correct?
Correct. And I don't know that we have a lot of choices anyway. I think streptomycin sulfate comes to mind, and it wasn't particularly effective. So
Tetracyclines, yeah.
So I don't think there's a lot of hope there, frankly, with Xylella. I think my thought on this has always been to get the tree to grow as good as you can so it can outgrow the bacteria. This is the more news. The real deal is in the last 2 annual growth rings. And if you can get those to be healthy by growing away from the pathogen, which is in the more interior rings, you can outgrow it. So doing things horticulturally that help the tree, whatever that means in your location, is the best thing to do to get out of that
Right?
disease. Yeah.
As we found with the way Xylella is affecting olives.
Yeah, it's a really weird disease in olive. And for me, I've struggled trying to understand if it even is a disease in olive. As it comes and goes, it gets better on its own, then it comes back really viciously. Botrosphaeria comes in and mimics it, so you don't know really what you got. And then there's been some disagreement in the literature about it. Some folks in Italy said, no, it's not Xylella, but then more recently, a lot of people said, yeah, Xylella is a really bad thing here. So there's some disagreement on all of what's going on. And why would that happen? Why would there be disagreement? Well, because the way you prove a disease is you isolate the pathogen and then you infect a healthy tree and you see if the same symptoms happen, and then you re-isolate in pure culture the pathogen again. And it turns out that's really hard with Xylella. The bacterium doesn't want to play in our Petri plates. And so it's hard to prove that disease, and therefore it's been hard to prove that Xylella actually is the cause of the disease in olive in a lot of cases, even though we think it is. I mean, you can PCR an olive and you get the pathogen out of it, so it's in there. But it's hard to do the other steps of Koch's postulates to prove it all.
So for fun, Michelle would like to know what our favorite trees are, yours and mine.
Ooh.
Ooh.
I guess for me, it's kind of evolving because I'm working with different trees lately, but I've always been fond of ginkgos, but I'm increasingly fond of oaks, particularly the Mexican ones. So I think maybe my favoriteest tree right now is Corkus Rugosa. How's that?
Is that red new growth?
Yeah, the red growth.
Nice.
It's in the last webinar.
That one, yeah. I'm not that familiar with that tree. I don't know that it's common in Southern California.
No, and it's one I want to be part of Southern California. I think it has some real promise for us. And the silver leaf oak, Quercus hypoleucoides, is also a very nice tree. So, and that is available, by the way, from Devil Mountain Nursery, also known as, what was the last nursery? The Bee Company.
The Bee Company?
And Valley Crest before that.
Oh, Brightview.
Brightview, yeah. Well, Brightview sold it to Devil Mountain, so they have Quercus hypoleucoides in stock, so you can get it. Well, Tracy, what's your favorite tree?
So I want to
You didn't say.
Emily's question first, because she has a more technical question. I had mentioned that one of the more susceptible trees to armillaria is Shinus malay, California. Well, I'd call it California, but it's Peruvian pepper. And
Correct
she's
name.
asking, removal. Is there any way to remove it? An arborist with a chainsaw? Or do you mean the armillaria?
Yeah. The best way to deal with that is with a backhoe. I guess you could go at it with a stump grinder, but the problem is the stump grinder is not going to get all the roots. So you would want to dig the stump out and get as much of the root system out as you can. And you can, while that's happening, you can see the fungus in the root system. You can look at it. It's like you can do your own biopsy as they go. Yeah, it's here. No, we don't have any over here. We're good. That kind of thing. But you have to it's physically removing the inoculum from the soil. And as far as I know, that's what a backhoe does, but not so much an arborist and not so much a stump grinder.
Yeah, it killed a couple of my aloes, which surprised me.
I've seen it kill bamboo, palms, and ferns, and begonias.
Yeah,
I've seen that. It just kills a lot of things.
I'd be surprised by. That's interesting. Yeah. My favorite tree is evolving also. Originally, I would have said Platanus racemosa, the California sycamore, but with
that's a good one
here, a well-grown California sycamore just looks amazing. But with the invasive shot hole borer, they just look like hell. So I'm and our drought conditions, because most people don't grow that riparian tree in the appropriate location or condition. So I'm less and less enamored with that. I absolutely love a well-grown Afrocarpus falcatus / Graciliar, because those can just be absolutely the most stunning tree, shade tree, glorious, strong, resistant in
tough.
my mind. Yeah, tough.
I've never seen one of those with armillaria. Have you?
Not to name drop, but microcritons struggled for a while because of, I think, the soil conditions. And we struggled with that in that location, but I don't think that tree ever succumbed. And it also struggled with other things. Never seen a well-grown Afrocarpus succumb.
You know, that turned into kind of a fad plant in my town. People were planting hedges of it.
Yeah,
They
it's
get 15-gallon ones and plant them 2 feet apart.
horrible, horrible.
It's like, what are they thinking?
Right?
Incredible, because what if that gets away from them, they're going to have like a problem.
Yeah. Well, they keep them hedged down, and they're those, stem cutting type grown ones, and I don't think that they have the trunk diameter or the energy. And often I'm seeing them planted in place of ficus as a hedge.
Right.
And the poorly grown, deeply planted ficus in many parts of LA will succumb to armillaria, where in other locations where they're well grown, I have seen them be quite tolerant. So personally, I would like research done on the impacts of growing stem rooted cuttings as opposed to growing trees that have a true root radically based root collar. I think that they may have different alterations around their newly developed root collar that may not thrive in the same way.
Okay, prospective students, sign up now.
Will grow. So, I mean, you may have completely different thoughts on that, and you might go,
The long-term study.
yeah, definitely long-term, but it just seems like, we're seeing a lot of these root collar depth issues on trees that have been grown from stem cuttings, and I'm curious about that. So, with that said,
it's 2 o'clock and I got a boogie, baby.
Question bank.
Thanks, everybody. Thanks for coming to the webinar series, and look forward to a survey coming soon.
Yay, surveys.